Hello, everyone, and welcome to this short talk on the century of the brain. My name is Dr. Thanasios Soulis. I am currently the program director of the MSc in Psychiatry at Cardiff University, and I also teach on the undergraduate medical program. I lead on the school's digital education strategy group, and most recently I've been appointed as the postgraduate taught studies co-director. But more importantly, this is a talk that I deliver as part of the School of Medicine's Science in Health event, which is hosted annually at Cardiff University. And the reason I've chosen this particular topic is because the brain is arguably the most complex of organs, and it has actually been described by some as probably the most complex structure in the known universe. And our understanding of the brain has increased exponentially over recent years. And the more we understand about the inner workings of the human brain, the more this will drive not only innovations in terms of better treatments that are tailored to individuals when we're looking at curing certain conditions, psychiatric and otherwise, but also it will drive innovation in regards to things such as artificial intelligence. But before we get into all of that, very, very basically, what is the brain? So the brain is this key organ that plays an important role in the most basic of functions, your ability to breathe, to remain conscious, to be aware of your surroundings. But it also plays a profound role in more sophisticated abilities. So your ability to think in abstract terms, to take in information from your environment and make sense of it, and even making you who you are as a person. So the brain actually plays a crucial role in not only who you are, but how you see yourself in the world around you. Everything that we are is dependent on this particular organ. Everything we are in the most basic sense, as well as the most complex. Now, when we're talking about the brain in its crudest form, we're looking at four basic lobes plus the brainstem. So we have, for instance, the frontal lobe, which is the part of the human brain that is evolved the most recently in our species. It's the home of our more sophisticated cognitive abilities, problem solving, abstract thought, and so forth. But it's also the part of the human brain that takes the longest to mature, only reaching full maturation during late adolescence, early adulthood. It's also the part of the brain that is the first to shrink in terms of gray matter volume, which accounts for the cognitive changes that we tend to see in the normal aging process as well. But there are other key lobes. The temporal lobe plays an important role in memory and learning, plays an important role in taking in auditory forms of information and making sense of those. The parietal lobe, amongst other things, plays an important role in your ability to navigate your way around your immediate environment. And the occipital lobe, crucial in terms of taking in visual information from your environment and making sense of that. Obviously, that's just a very, very brief overview, much more complex mechanisms underpinning each of these lobes. But for the interests of this particular talk, we're just going to cover the very, very basics. The brainstem, divided into three further parts, plays an important role in, for instance, as we said previously, breathing, conscious awareness of your immediate environment, and other basic functions as well. There's also the cerebellum, which is not really on this particular slide, interestingly enough, but the cerebellum plays an important role. It's that sort of cauliflower-like structure, sort of just underneath the occipital lobe, and that plays an important role in, amongst, again, other things, coordination of movement and so forth. And what's important to note here as well is that our understanding of what these lobes and the structures within these lobes actually do is increasing day by day. We're understanding a bit more about the inner workings of these lobes of the brain as a whole, the structures, the circuits, and the various networks. And our knowledge is also open to being built upon in a way that may make us reflect on previous findings and reconsider what we actually thought certain parts of the brain actually do. And we're not only looking, for instance, at what the brain is in terms of gray matter volume, but we're also interested in what we refer to as white matter and the white matter tracts, those parts, that part of the brain that connects one structure to the other, and it enables the brain and the various structures in the brain to communicate with one another. But before we get on to that, I want to introduce you to the case of Phineas Gage. Many of you may be familiar with the story, or at least have heard of Phineas Gage. But it's an interesting case study because Phineas was someone who revolutionized our understanding of the inner workings of the human brain earlier on in the 19th century. So he was a railroad foreman working on the construction of railroad tracks in the United States, 19th century. And on one fateful day, as legend has it, he was overseeing the inserting of gunpowder in the surrounding bedrock that they were going to blast away so that they could install the railroad tracks in that particular part of the country. So he was using the tamping iron that you see him holding in this image, and he was using that to actually insert the gunpowder into the ground. Now, the story goes that he was distracted whilst doing this, and the tamping iron struck a nearby rock, igniting the gunpowder and effectively turning this tamping iron into a missile, which penetrated his cheek, went through his head, and landed a few feet away from him. Again, as legend goes, when the town's physician was called to the scene, Phineas was sitting upright and according to accounts, was engaging with the town's physician. Some accounts actually refer to him saying something along the lines of, well, how do you explain this? And I may have misquoted there, but it's something along those lines. Now, why would someone who had a tamping iron basically go through his head survive such an ordeal? And that's a really interesting question. So as you can see, that is the sort of environment he was working in, and clearing the rocks in the surrounding area by blasting them away. But that tamping iron actually caused him severe brain damage. And based on subsequent analysis following his death of where the damage appeared to occur when looking at his skull, what we suspect happened is that the tamping iron penetrated a part of the frontal lobe that we refer to as the orbitofrontal cortex, as well as obviously doing some damage to the surrounding structures. Now, our understanding of the orbitofrontal cortex is that it plays an important role in things such as reward and motivation, but it also plays an important role in personality and various other complex abilities and functions as well. And this is consistent with reports of how Phineas changed following the incident. So his family, his friends didn't report any profound effect on his cognitive abilities or memory. There may have been some effects there, but most notably, his friends described him as no longer being Phineas. He had changed, his personality had changed. He also, his responses appeared to be more childlike in terms of immediate gratification and so forth. And that told us something very, very important. Damage to one particular part of the brain can actually have a very, very profound effect on a person's ability to function, in this instance, socially. So Phineas Gage was a pivotal case study in our understanding of the human brain. He was actually brought into Harvard University, seated in the front of a lecture theatre, and used as a case study to medical students at the time of basically, how do you explain this? Now, when we're talking about various structures in the brain, we tend to focus on gray matter. But research currently is looking more so at the role of white matter tracts, white matter tract integrity, and what all of this means in terms of not only psychiatric illness, but even the aging process and pathological forms of aging as well. When we're looking at things like Alzheimer's disease and other dementias. And the work that we're currently looking at is basically, and when I say we, I'm talking collectively, this is not specifically my research, but what is of interest in this particular area is how these white matter tracts actually also contribute to our ability to perform our day-to-day activities and the basic functions of the human brain, both in terms of the complex and the not so complex. And what we know is that also focusing on these specific, or rather these specific tracts, what we also should consider is how intervention and treatments may also be designed in such a way as to look at improving the structural damage, perhaps, that has occurred in parts of the brain, not just when we're focusing on certain lobes and structures in those lobes, but also the white matter tracts that sort of lie beneath all of that. So there's a lot of research currently taking place in this area, but in all honesty, there's a tremendous amount of research taking place in neuropsychology, neuroscience, and psychiatry in general, looking at the genetics and genomics underpinning various psychiatric conditions as well, and why there are certain structural changes to the brain in patients who present with various psychiatric conditions, such as early onset psychosis and so forth. So the second case study I want to present very briefly is that of Henry Molaison, and some of you may be familiar with this case as well. Now, Henry was someone who lived in the 20th century. He played a pivotal role in enhancing our understanding of human cognition and memory specifically, because Henry, unfortunately, had undergone surgical procedure that had a profound impact on his long-term memory. So at the age of around 10 or 11, Henry was experiencing very debilitating epileptic seizures, and his team of physicians and surgeons decided at the time that the best course of action for Henry was the complete removal of the hippocampus. So hippocampus and surrounding structures, which they hypothesized, they thought, would relieve Henry of his very severe seizures. Now, the good news is that complete removal of the hippocampus did have an impact on Henry's seizures. It did relieve him of those very debilitating seizures that he had been experiencing in his early life. But in the 1950s, our understanding of the human brain wasn't all that sophisticated. What his team of physicians and surgeons hadn't considered was that the hippocampus perhaps plays an important role in other things as well. And what we discovered in Henry's case was that it plays a profound role in long-term memory formation, specific types of long-term memory as well that involve memories that are of a personal nature to you, as well as long-term memories about the world around you and information pertaining to your immediate and not so immediate environments, i.e. kind of general knowledge type of information. And that was interesting. Very sad in Henry's case. What they found in Henry's case was that his short-term memory, his working memory, remained intact, but he was stuck in this kind of limbo, this purgatory of only remembering up to maximum 30 seconds to a minute. Beyond that, information was completely lost because of the removal of the hippocampus and disrupting that particular memory network, which did not allow for the creation and the formation of new long-term memories. So Henry was actually unable to form new long-term memories. It was a severe and pervasive form of amnesia that he lived with for the rest of his days. So profound and so debilitating was his condition that he was interviewed at times for years on end by specific researchers who visited him, and he didn't remember meeting those particular individuals the day before, even though this happened in one particular instance for a decade or more. The surgeon who led on the procedure, which affected Henry's memory, Scoville, wanted to also remain involved in Henry's case because he wants to ensure that this same kind of event didn't occur again. No one else had to experience what Henry was going through. And Henry described his state as being sort of a dreamlike state, very, very uncertain experience of his surroundings and his environment. Henry lived to the ripe old age of 82, but he was always stuck in that period of short-term working memory. He could not form any new long-term memories following this procedure. Upon his death, Henry's brain was actually cryogenically stored, it was frozen, and consent was given from the family for it to be studied for research purposes, looking further into the damage that had occurred as a result of the procedure. So we knew what parts of the brain had been removed, but were there perhaps incisions or accidental damage to other structures that may have also accounted for certain things that were observed in Henry's cognitive abilities following his following the procedure itself. The brain itself was subsequently sliced into really, really thin slices, and it has been stored at a university, I believe, in California. The slices have been digitized and stored. They form a very, very important part in our understanding of the human brain and what specifically the parts damaged in Henry's case actually do in terms of memory and cognition. And there you see how the brain was actually stored and plays a profound role even to this day in our understanding of memory and cognition. Now, this is a video which is available, I believe, on YouTube, so I'm not going to show it right here, but I remember being a PhD student and actually watching live, they were broadcasting this live, the slicing of Henry's brain, which when I think back to it is a little bit weird. But it was pretty much because when my peers and I were in university, this was a case that we were pretty much familiar with, and it was quite an event to actually watch this great gift to science being donated and being sliced so it could be digitized and stored for future generations as well. And finally, the last case study I want to touch on is that of Roald Dahl. So hopefully everyone is familiar with who Roald Dahl is, but we're not going to talk about any of his literary works. We're going to talk about his role in neuroscience, which many people may not be aware of. And here you see Roald Dahl with one of his collaborators, and I'll talk about the instrument that his collaborator is actually holding there. So Roald Dahl was no stranger to head injury, neurology, and neuroscience. He was a fighter pilot in the Second World War. When his plane was damaged, when he crashed, he suffered brain injury, and he actually jokingly attributed his creativity to that incident. We take that with a bit of salt. But he also experienced a couple of other episodes where in his family life where he actually played an important role in our understanding of the human brain. So the first instance was one of his children. So many people are aware that one of Roald Dahl's daughters sadly passed away of measles, but he also had a son who, as an infant, had hydrocephalus. And what Roald Dahl played a crucial role in was developing, alongside researchers and a tool manufacturer, a particular shunt, a valve that played a really, really important role at the time in relieving the pressure in the heads of, for instance, his son, his infants, and other patients as well. This shunt was actually used elsewhere and had been used hundreds of times, and it was thanks to Roald Dahl and his collaborators for actually creating this particular instrument. I believe it's known as the Wade-Dahl-Till valve. I'm not sure if it's still used today. I'm sure we have instruments that are probably more sophisticated, but very, very revolutionary, a pioneering piece of work for its time. And here you can see how important that particular instrument was in the personal life of Roald Dahl. But in addition, this was, again, a great gift to humanity. He also played an important role in the recovery that his wife went through following a stroke. So rehabilitation following her stroke. At the time, the rehabilitation program was not what it is today. And what it is today is very much attributed to the important work that Roald Dahl did in pushing various reforms and a different way of actually thinking about stroke rehabilitation. He wanted to help his wife return to as normal a life as possible, and he played an important role in helping her in that regard. And his approach was actually very inspirational. He did play a pioneering role in that area as well. So again, there's an example, there's an image rather, of the Wade-Dahl-Till valve, which has been used. It's a cerebral shunt, and it was used at the time to relieve pressure in the as we mentioned previously, in the cerebral edema in patients who had, for instance, hydrocephalus. So ending off, just thinking about what does all this lead us to now. So obviously the case studies I've presented are quite old, but what we're looking at now are kind of the really, really exciting findings that are emerging in the research over the past few decades and even the past few years. Again, this area of research is growing so rapidly. And at Cardiff University, we have excellent research teams in the Hayden Ellis Building, Division of Psychological Medicine and Clinical Neurosciences. Incredible teams undertaking incredible work in conditions such as psychosis, the dementias, in terms of neurodevelopmental disorders, and so forth. And next door to Hayden Ellis is the Cardiff University Brain Research Imaging Center, Kubrick, which has some of the most sophisticated machines at our disposal, 7 Tesla, and really, really powerful magnets that enable us to get a better look in relation to the inner workings of the human mind. So when patients are undertaking, for instance, behavioral or cognitive tasks, we can actually get a better idea of what's happening in the brain and compare one group of patients with another. And the scans and the findings, the results that are published actually enable us to push the boundaries of our scientific understanding. And that also influences the application thereof when it comes to treatments for various conditions, psychiatric as well as non-psychiatric, and the application of that knowledge to other exciting areas as well. And as mentioned previously, we're also interested in looking at the brain as a whole, not just lobes and structures and gray matter, but white matter, the various cells that play an important role in gray matter and white matter, and how all of this creates a picture that improves our understanding of what the human brain actually does, and when something goes wrong, where does this go wrong, and how does that impact, for instance, the presentation of a particular condition. And another area that is growing rapidly, again, Cardiff University is a pioneer in this research, is genetics and genomics. Some really, really exciting research in that area, specifically looking at research groups focusing on psychosis, schizophrenia, and so forth. And our understanding of the genetic basis of many of these conditions is playing a profound role in improving our understanding of the etiology, the causes that give rise to these conditions. So we know that there is a prominent biological role. We know that there is a prominent environmental role. And it's not about nature versus nurture. It's about how the two come together to predispose certain individuals to these particular conditions. And the neurobiology, the genetics, these play a profound role in that as well. It's not just environment, and equally, it's not just the brain, but the genetic component here is something that we're understanding in a lot more detail. It not only plays an important role in our understanding of those conditions, but also how we can develop more tailored treatments for patients in the future. So that leads us finally on to the last slide. What is the future? Again, tailored treatments. Currently, we have medications, for instance, antidepressants, antipsychotics, that perhaps work for a good proportion of patients, but they don't work as well for everyone. And it's important to develop new drugs, new medications based on new technology that enables us to tailor these approaches and ensure that we're not only treating patients, but where possible, curing them of these debilitating conditions as well. Artificial intelligence is another area that's growing rapidly. So we know that there's a lot of talk about merging humans with machines, probably a little way from that now. I think we've only just managed to get robots to stand upright as opposed to just falling down. But even something like that demonstrates how powerful the human brain is. Your ability to stand upright and walk in a straight line, we take for granted. But trying to get a robot to do that, the research suggests it took some time to get them to do that. And that's not even looking at the more sophisticated abilities that we possess. But there are, for instance, when we're looking at machine learning, various platforms out there that demonstrate how powerful machine learning can be. For instance, ChatGPT can apparently write your essay for you. I wouldn't recommend that though, because there is work currently taking place to demonstrate how we can trip ChatGPT up. But what I think is important is that as we look into the future, whether it's medicine, artificial intelligence, what have you, as long as we're using these advances to improve the human condition, to make life better for the majority of people on the planet and for the planet as a whole, then we've achieved our goal. And the generations come after us, they are the ones who are going to be leading this work. They may be correcting some of the findings that we currently may believe are an accurate representation of the natural world. And that's what science is, building on the knowledge of others, perhaps filling in gaps or replacing knowledge with new knowledge. And in doing so, using that to improve life for the people who we share this planet with. So that is my few minutes of talking about the center of the human brain. I hope you found it interesting. If you have any questions, please do get in touch. Thank you.